Hello, everyone. My name is Lindsay Clontz, and I'm a master's student at the University of Georgia in the Warnell School of Forestry and Natural Resources. And today I'm going to be sharing with you some research I've been doing on behavioral state resource selection of wild pigs in the Southeast United States. So as the most introduced ungulate in the United States, wild pigs have a widespread and noticeable impact, including they degrade agricultural and natural areas such as harvestable crops, streams, wetlands, etc. They also predate and compete with native species, as well as transmit diseases to other wildlife species, domestic livestock, and people. It's estimated that every year wild pigs cause approximately $800 million worth of damage in the agricultural sector alone. However, we realize this is a conservative estimate overall because it's difficult to put a monetary value on something like transmitting a zoonotic disease or causing irreparable damage to natural areas. So we see that wild pigs are a bit of a paradox because they're associated with all this negative press, but in the same light, they're a prized game species that many people's hunting traditions revolve around, and other people are really eager to attempt some of these traditions. So this creates a really unique situation for managers when we're trying to come up with management programs and management techniques to control invading populations and growing populations across their invasive range. And we realize that our current management strategies right now are insufficient and that we need more tools to better control wild pig populations. So as we think about how to manage wild pigs, we think about what they're doing, how they're interacting with resources across the landscape, and what all this means. Compared to other ungulates, wild pigs are really understudied in the world of spatial ecology and how they select resources associated with common behaviors. And given their expansive range, it's really important for us to understand these intricacies of how they interact with different landscape features and vegetation types. So we have defined a behavioral state as just a response to an environment. And we realize that these behaviors can actually be associated with resources that are distributed heterogeneously across the landscape. And therefore, we see this in turn driving animal movement. For example, wild pigs have difficulty thermoregulating. So a lot of times we find them in areas proximal to water or in dense cover, which ultimately we can read as something that they're interacting with this specific environment to help them act out a certain behavior or something that's necessary for them to do. And we see that resource availability fluctuates throughout the year, especially in the Southeast, and we see how resource selection changes for wild pigs and patterns of this change throughout the year in return of these resources changing. And so we see all these behavioral decisions being pretty much at the heart of all these resource selection questions, and yet they've been ignored in management mainly because we've lacked the technology to get at some of these fine-scale questions. But now that we have stuff like GPS technology, we can answer some of these fine-scale questions. So this project is ultimately trying to explore the correlation between wild pig behavioral states and resource selection across two distinct seasons based on forage availability. This study was done on the Savannah River site, which is approximately an 800 square kilometer site managed by the Department of Energy. It has a very high density of wild pigs and a very diverse set of landscape features and vegetation types, with less than 5% of it being characterized as developed. As you can see on the right-hand side of your screen, we have like several collared pigs that expand practically our entire study area of the SRFs. So these animals were trapped from January 2014 through December 2019 using corral traps with both manual and electronic gates. And we used a combination of either BAM and ketamine or xylazine and telazol to anesthetize all our wild pigs. And we placed GPS collars on all dominant females, which were identified using remote camera surveys throughout this study, and all breeding age males. We then reversed anesthesia and released animals back at the trap site for tracking. We identified two distinct seasons in the Southeast based on wild pig activity patterns, as well as unpublished diet data from the Savannah River site. In ways that we felt best represented our collared animals. So we defined the low forage season from January through April as a food scarcity season, where wild pigs mainly focus on subterranean foods such as roots, tubers, earthworms, grubworms, stuff like that. And then we had the high forage season, which is a really productive time in the southeast, which is from late spring or May through early winter, December. And this time has stuff like, or is characterized by fruits such as blackberries, plums, productive plants, and probably the most important, oak acorn or other hard mast and nuts. So during the low forage season, we had 37 individuals, 21 of which were females and 16 were males. And then during the high forage season, we had 41 individuals, 20 of which were females and 21 were males. And all of these pigs were equipped with a collar that captured a GPS location every 1 hour. You can see there's some discrepancies in our sample size across seasons. Most of these wild pigs did span across seasons. However, we did have animals that slipped collars or expired throughout our season or throughout our study, and those animals were replaced, and that's why some of our samples are different between seasons. We used hidden Markov models to identify our behaviors for our study. Hidden Markov models are made up of two processes, an observation or parameter process and a hidden state process. The observation and parameter process is the informative process of this model. And we use step lengths and turn angles, or basically movement characteristics, to inform our model. This model gives us an initial distribution, which is ultimately any given time what behavior they would be in, and then also a transition probability, which tells us what's the probability of them staying in a state versus transitioning to another state. And like I said, we used these step lengths and turn angles to basically identify nodes in our data that showed us biologically relevant behaviors that we could pick out and therefore characterize with this model. So we chose to characterize resting, which was short step lengths and tight turn angles, foraging, which was characterized by short to medium step lengths and tight turn angles, and traveling, which was longer step lengths and shallow turn angles. So we fit this three-state hidden Markov model to these three behavioral states that we were able to identify for both males and females separately for each season. And then we used time of day as an additive covariate in our models. We ran 25 iterations of these hidden Markov models to identify or to make sure we had numerical stability and we're identifying global maximums for each behavior instead of local maximums. And then we used the Viterbi algorithm to decode behavior for each location or each individual GPS location for all our collared pigs. After that, we generated covariate layers using national land cover data from 2016, as well as some other Savannah River site generated layers. After identifying some of these vegetation types and isolating them, we used the Euclidean distance tool in ArcMap to get a distance 2 value for each one of these vegetation types or landscape features. And we also had the National Land Cover tree canopy data from 2019 to get percent canopy cover as well. After that, we generated 95% kernel utilization distributions for all individuals in each season. And then we systematically sampled these home ranges with for every 1 point every 90 meters, which was sufficient to represent available habitat in our study area. We then separated locations by sex, season, and behavioral state for modeling purposes, and we used the same available locations for each sex within each season. We then created our generalized linear mixed models. We did a global model for both sexes in each behavioral state in all seasons, with a random intercept for individual to account for differing or unbalanced sample size of GPS locations among individuals. And we did not use any kind of model selection technique, so it would allow for a direct comparison across beta coefficients for both sex, season, and behavioral state. So for our results for our distribution of our parameters from our hidden Markov models, like I had said, we had characterized 3 behavioral states, and you can see the 3 behavioral states defined, much of like I showed in the animation before. So we had the resting state, which was defined by these short step lengths and these tight turn angles. We had the foraging defined by these short to intermediate step lengths and these tight turn angles as well on the bottom. And then we had traveling defined by these longer step lengths and these shallow turn angles around 0° radians. And so this is for low foraging season, and we had separated our seasons, but this is high foraging season, and we see much of a very similar distribution of our states, where we know that we have identified at least 3 biologically relevant states that take up approximately 90% of wild pig time. These graphs are depicting the proportion of behavioral state per hour across the diel period. We have females in the low and high forage seasoning on the left and males on the right. The dark gray bars represent nighttime hours and the light gray bar represents daytime hours. So in the low forage season, we can see that females have a very variable activity pattern. And actually this low forage season in the southeast is directly compared to a reproductive peak for wild pigs during this time. So we expect that some of this variability is related to that. But we can see that they have a definite peak at dusk in their traveling, as well as they tend to travel and forage much throughout the daytime, while they are resting more at dawn and some throughout the night. But we see in the high forage season, they have a very clear crepuscular pattern where they are traveling much at dawn and dusk, but they are foraging throughout the night, and then some in the, or they're foraging pretty similar to when they're traveling, but they're resting throughout the night and in the midday. And then we see males have a very consistent pattern across both seasons, where they are traveling throughout the nighttime hours and resting mainly during the daytime hours. For our resource selection, our behavior resource selection in the low forage season, I'm just going to point out some of the main points. To the left-hand side of the dotted line will be selection for all of our distance 2 variables, and for canopy cover, it'll be to the right-hand side, and it'll be inverse for our avoidance. So we'll have avoidance on the right-hand side for all our distance 2 variables and avoidance for canopy cover on the left-hand side. So one of the main things is this proximity to streams. Females actually avoided areas that were closer to streams while resting, while males selected for areas closer to streams while resting. And females actually selected for areas in the shrub herbaceous type community, which is characterized by primary and secondary succession along usually power lines, secondary roads, streams, and other places. They selected for this area while resting, but avoided this area while foraging and traveling, while males actually selected for this area throughout all behavioral states. The distance to secondary road, females avoided areas close to secondary roads while resting and foraging, while males selected for areas close to secondary road while foraging and traveling. And then I have these highlighted in red for both males and females, because consistently we see they selected for areas close to bottomland hardwoods or within bottomland hardwoods, and areas close to high percentages of canopy cover in both, or in all behavioral states in the low forage season. During the high forage season, we see a few differences between males and females, as well as from the low forage season. Females show a affinity for upland hardwoods in all behavioral states, while males actually avoided upland hardwoods in the high forage season while foraging, but selected for this while resting. Once again, we see this distance to stream is avoided by females during all behavioral states, but it's and it was also avoided by males during all behavioral states. And then secondary road, once again, is avoided by females during all behavioral states, but is avoided for males while resting, or resting and foraging, but is selected while traveling. And then lastly, we have this still consistent behavior of where wild pigs are selecting for areas close to bottomland hardwoods and areas of dense canopy cover. We see several similarities and differences demonstrated between sexes and across seasons as far as this behavioral state resource selection goes. For example, one of the big ones is males demonstrated a very a variable foraging pattern in the low forage season compared to solely selecting for bottomland hardwoods while foraging in the high forage season. We expect this is most likely due to resource availability, or males are targeting acorns and other productive understory plants in the high forage season in these bottomland hardwoods, while they're looking at anthropogenic sources and other sources for food during the low forage season, such as primary roads, secondary roads, and other places that might have some productivity in the low forage season. Also, we see that males and females have solely selected for, or have selected for bottomland hardwoods in areas of high percentages of canopy covers consistently throughout both seasons. And we suspect this is most likely due to these areas are proximal to water. They are also have dense cover, and they're associated with forage types throughout both low forage and high forage seasons. We see that generally at a broad scale, when we're looking that males and females may select for areas close to streams or areas close to upland pines and stuff like that, that may not be actually showing up in these fine scale results, because at a fine scale, pigs are drilling down and selecting for key resources, even though they might be a generalist and they're ubiquitous usually throughout the entire Southeast. We see that there are specific things that are important to wild pigs that we can identify as key resources that can potentially help us as managers focus in on these areas to target and control wild pigs better. So with that being considered, without defining behavioral states and separating them into these separate analyses, we could see that a lot of this stuff might be washed out if we did a general RSF with all of our locations, where we picked up on male wild pigs traveling, only selecting secondary roads while traveling, but avoiding secondary roads while resting or foraging, or females resting in shrub and herbaceous communities, but avoiding these while in the foraging and resting, or foraging and traveling states. We might have missed some of this stuff if we would have done just a general RSF. So actually looking at data very fine scale can actually help us target wild pigs better. And then also, I'd like to point out that wild pigs are very unique in their reproductive abilities, and therefore we're unable to control and look at each reproductive stage for females. However, we realize there's most likely a difference between behaviors and resource selection for each reproductive stage for females, since they have so many reproductive responsibilities in wild pigs. So we think that it would be really interesting for further research to target some of these different reproductive stages for females, as far as prior to conceiving, as well as through pregnancy and after that in the parental care type stages, to see how their behaviors are changing and how resource selection changes throughout those stages. Lastly, I'd like to talk a little bit about management implications of this. We hope that some of this fine scale data can help us target pigs when they're most vulnerable, whether that be when they're most active or in areas where they maybe spend more time resting and foraging versus where they might be a little more variable when they're traveling. But we can take certain management techniques, whether it's toxicant deployment or aerial gunning or hunting or whatever it might be, and try to match that with a specific habitat type and specific time of day that we think might fit the best with that management technique, as far as what we know about wild pigs at this fine scale. Also, we would like to just contribute and hope that this kind of lays a foundation for the literature to continue to build on some of these fine scale questions to help us potentially target wild pigs in more of a fine scale way, as well as ultimately decrease the negative environmental and economic impacts of wild pigs. So with that, I would like to thank some people and acknowledge several organizations. The funding source was USDA APHIS Wildlife Services, as well as I'd like to thank the University of Georgia and the Savannah River Ecology Lab, as well as US Forest Service for helping with some of the trapping, US Department of Energy, and then several colleagues and project technicians that made this project possible. With that, you can either comment on this presentation with questions or email and provide questions, and I would be happy to answer those.